This video provides brief summary on slides related to the presentation given by me on the topic promoting healthy aging and longevity. The presentation details insights from analysis of large-scale public health data of individuals across lifespan using statistics and artificial intelligence tools. It also briefly covers molecular mechanisms behind aging and healthy lifestyle. My goal is to interact with individuals passionate about tracking their biomarkers and support collection of de-identified data to improve precision care and motivate healthy habits. Preview. Aging is a universal phenomenon involving physical decline from the cellular to systemic levels. Chronological age does not accurately reflect an individual's actual biological health, and aging is considered a major risk factor for many chronic diseases. Talk outline. The presentation covers defining and measuring biological age using biomarkers, how public data and AI provide insights on tracking chronic health disease risk, and proposes a mission for healthy aging. Research on science of aging. Brief overview. Highlights core longevity research concepts including caloric restriction, telomere attrition, cellular senescence, epigenetic clocks, hallmark of aging, etc. Phenotypic age. This slide shows the article that details the main metric called phenotypic age used for measuring biological age covered in this presentation. Multiple methods can be used to measure biological age with the different sets of clinical biomarkers. The slide highlights one such article that details application of deep neural networks for biological age measurement. Phenotypic age, very practical tool for measuring biological age. A simple mathematical formula and computer code is sufficient to calculate phenotypic age and 10-year mortality risk directly from routine lab results. Comparing and correlating biological age. The slide presents a data table that compares estimated biological age against chronological age and shows how specific lab markers influence biological age in different individuals. Age-associated changes in biomarker level over human lifespan. This visualizes how average values of certain biomarkers changes with the age when observed over large-scale population. Displaying graphs where mean fasting glucose increases and serum albumin decreases as people get older. Age-associated changes in biomarker level over human lifespan. NHANES data. The slide provides further scatter plots demonstrating how markers like lymphocyte percent and serum creatinine level correlate with the chronological age and shows the findings can be easily reproduced using massive public dataset. NHANES Public Health Survey. The slide introduces the CDC's National Health and Nutrition Examination Survey, the primary source of the roughly 100,000 data used for the analysis. Hazard ratios. Displays graphs showing that mortality risk hazard ratio steadily increases as serum glucose levels deviate from an optimal baseline range. Phenotypic age, chronological age, and phenoage Excel. The slide illustrates a strong correlation between phenotypic age and chronological age and introduces the concept of phenotypic age acceleration, called simply as phenoage Excel. Measurement of phenotypic age directly from NHANES data. Shows additional scatter plot and the histogram representing standardized measurement of phenotypic age acceleration derived from the public health data. Kaplan-Meier curves for persons in the highest 20% versus the lowest 20% of phenoage Excel. Demonstrates that individuals in the top 20% of phenotypic age acceleration have noticeably lower survival rates over follow-up years compared to those in the lowest 20%. People do track the biological age regularly. Presents a mock tracking chart showing an individual's effort to keep their measured phenotypic age consistently lower than chronological age. mean values of analyzed NHANES samples. Provides the statistical means and standard deviation for various biomarkers from the data analysis alongside their standard normal ranges. Comparing lowest and highest phenoage age quartile. Compares the raw clinical data profiles of people in the lowest versus highest quartiles of phenotypic age, revealing vast differences in metrics like glucose and high-sensitivity CRP. Impact of body mass age on phenoage. The slide shows the correlation between BMI levels and phenotypic age acceleration and a plot of a linear trend line. Comparing biological age biomarkers within and across different BMI groups. Shows that biological age is not strictly tied to weight, noting that some normal weight individuals exhibit a higher biological age than chronological age, while some obese individuals exhibit the reverse. BMI versus phenotypic age relation is complex. Cites research suggesting that once fasting insulin and high-sensitivity C-reactive protein are accounted for, high BMI level alone does not increase mortality, indicating that hyperinsulinemia and inflammation are the true drivers. AI-based phenoage Excel prediction uses an AI model to rank the most important features driving age acceleration, identifying factors like RBC distribution width, fasting glucose, and serum creatinine as highly influential. SHAP importance for phenotypic age estimation features for different population heterogeneity. Uses SHAP plots to demonstrate how specific biomarkers drive biological age estimates differently, depending on population traits such as body mass index. SHAP importance major tool to identify important biomarkers from ML models. The slide highlights the SHAP tool as a crucial method for interpreting machine learning models and ranking the relative importance of biomarkers. Age-associated increase in homocysteine level across lifespan. Provides a scatter plot demonstrating a positive correlation between advancing chronological age and an increase in blood homocysteine levels. Factors influencing homocysteine levels and the level's impact on various organ systems. Features an infographic detailing the numerous lifestyle, genetic, and medical factors that impact total homocysteine level, and shows how chronic high levels of homocysteine impact various organ systems. Homocysteine blood biomarker metabolic pathway. Explores the complex metabolic pathways connecting homocysteine, folate, and other B vitamins. Feature importance SHAP summary tool for homocysteine. Uses SHAP analysis to reveal that a person's age, serum creatinine, folate, and vitamin B12 levels are the strongest AI predictors for determining their homocysteine levels, reinforcing age-associated change in levels. Feature importance SHAP summary plot predicting factors influencing biomarkers. Shows SHAP analysis predicting what physical factors drive specific biomarkers like insulin and high-sensitivity CRP, heavily implicating BMI and the waist circumference for insulin and BMI and WBC count for hsCRP. Summary phenotypic age. The slide summarizes that single biomarkers exhibit clear age-associated changes. The longevity biomarkers influence all-cause mortality, and the AI tools like SHAP are highly effective at identifying biomarker differences across various population subgroups, and major factors influencing levels of a given biomarker. Promoting Healthy Aging and Longevity Part 2. This introduces the next part of presentation. The impact of aging-related changes on chronic diseases and predicting chronic disease risk. Diabetes risk factors. This slide highlights multiple risk factors for diabetes such as defective insulin production, impaired insulin secretion, and insulin resistance in multiple tissues. Longevity biomarkers have strong correlation with the clinically relevant biomarkers for chronic diseases. This slide argues managing the longevity biomarkers may reduce chronic diseases, showing data on how markers like serum alkaline phosphatase correlate with the diabetes prevalence and fasting glucose levels. Chronic inflammation impact on blood glucose level. This slide presents scatter plots and charts demonstrating a positive correlation between chronic inflammation measured by hsCRP and the blood glucose levels. Age-associated increase in disease prevalence. This asks whether preclinical biomarker changes occur before clinical ones, visualizing how the prevalence of diabetes increases with the age even when biomarker levels are fixed, as shown for serum alkaline phosphatase levels and the triglyceride levels. Insulin versus glucose and BMI levels. This slide shows insulin has an independent association with the body mass separate from diabetes risk, noting that individuals with a low BMI can have a higher risk of diabetes than those with a high BMI for the exact same insulin level. Average blood sugar level for different value ranges of BMI and insulin. This uses heat maps to explore whether insulin levels influence blood glucose levels when controlling for a specific BMI range from normal to morbidly obese. Average blood sugar level for different value ranges of triglyceride and insulin. This demonstrates that triglyceride and insulin levels are correlated, and it uses heat maps to show their synergistic role in influencing average blood glucose and disease prevalence. Multiple risk factors play synergistic role. This slide emphasizes that multiple risk factors such as hsCRP, insulin, and triglycerides work together synergistically to increase the overall prevalence of diabetes. AI model prediction risk of getting diabetes. This outlines a random forest machine learning model that takes inputs like age, BMI, insulin, WBC count to make predictions such as blood glucose level, HbA1c levels, or risk value for diabetes. Feature importance sharp summary plot predicting factors influencing diseases and disease-associated complications. This uses sharp plots to rank which factors have the highest average impact on AI models' output, such as age and insulin for HbA1c prediction and serum creatinine for the urine albumin-creatinine ratio. Feature importance for predicting glycosylated hemoglobin or HbA1c on individual basis. This visualize shows how specific risk factors like BMI, waist circumference, and age interact to determine the HbA1c levels for a specific individual. Dementia risk factors. This slide displays the age-related decline in right hippocampus volume and maps out modifiable dementia risk factors across early, mid, and late life. Aging process may resemble the pattern of many chronic diseases. This suggests the trajectory of aging mirrors chronic disease, illustrating that molecular level changes happen decades earlier before structural and functional changes manifest. Prediction accuracy to identify mild cognitive impairment from cognitive normal individuals. This compares confusion matrices to show the model's accuracy in predicting mild cognitive impairment versus normal cognition, both with and without cognitive assessment data. Feature importance sharp summary plot MCI risk prediction. This details the top features for predicting MCI risk, highlighting cognitive assessments like RAVELT when they are included and the biological markers like FDG and hippocampus volume when cognitive tests are excluded. Role of metabolomics. More precise insight at pathway level. This introduces metabolomics for pathway-level insights examining acetylcarnitines for a fatty acid oxidation, phospholipids, branch-chain amino acids, and omega-3 omega-6 ratios. Novel biomarkers for prediabetes identified by metabolomics. This presents a complex pathway diagram showing how specific metabolites, like acetylcarnitine C2, glycine, and the LPC, interact with the enzymes and the type 2 diabetes-related genes. Association between circulating blood metabolites and the brain amyloid beta deposition in Alzheimer's disease. This highlights research connecting specific circulating metabolites directly to amyloid beta deposition in brain. Interventions for healthy lifespan. This slide lists key lifestyle interventions including exercise, managing body mass index, getting enough sleep, reducing stress, and the consuming a balanced diet rich in fiber and monounsaturated fatty acid-rich oils like olive oils and omega-3s. Review, summary, and future mission. Transition slide marking the beginning of the presentation's concluding summary and the future outlook. Summary 1. This emphasizes the importance of observing biomarker trends early for better prognostic value, noting that multiple risk factors usually influence aging and chronic disease. Summary 2. This notes that longevity biomarkers helpful for assessing disease risk, like hsCRP, are often different from the biomarkers used for making clinically actionable diagnostic decisions like blood pressure or HbA1c. Summary 3. This explains that there are often no hard cutoff limits for many longevity biomarkers and the cautions that while biomarkers are correlated, they may not be causally related. Summary 4. This notes that tracking lifestyle choices alongside biomarker changes provides clues on how to improve health and lists additional biomarkers of interest like glutathione, sleep, heart rate, neutrophil to lymphocyte ratio, etc. Understanding molecular mechanisms behind aging and chronic disease development. This displays a diagram illustrating how SIRT2 regulates NLRP3 inflammasome activation, which is linked to inflammation and insulin resistance during aging. The slide also displays molecular mechanisms behind exercise-induced tissue regeneration. This slide explains the mechanism by which monounsaturated fatty acids like those in olive oil activate SIRT1 via PLIN5, emphasizing that this signaling, which is beneficial, only occurs when the fat is broken down during exercise or fasting. Quantitative mathematical modeling. This presents differential equations and the time series graphs used to mathematically model the dynamics of glucose and insulin over time. Carbohydrate transport cycle. This features a detailed diagram mapping how glucose is stored, oxidized, and transported between the gut, liver, blood, muscle, and the adipose tissue during both fed and fasted states. Lipid transport cycle. This displays a comprehensive flowchart tracking the spatial and temporal dynamics of lipid storage, oxidation, and transport across different tissues. Useful for building system dynamic models to capture complex interactions. Mission. This outlines my aim to interact with individuals passionate about tracking their biomarkers, aiming to collect de-identified data to improve precision care and motivate healthy habits. Further consideration. This identifies longevity science and multiomics as emerging fields advocating for further research, interdisciplinary interaction, and funding. Capitalism and the wellness gap. This critiques profit-driven healthcare for focusing on pharmaceutical symptom management rather than preventative neuroscience and argues that economic stress forces people into habits that accelerates biological aging. Disclaimer. The presentation's findings are preliminary. The objective is to provide a framework rather than treatment, and individuals should consult physicians for medical advice.